So what is the definition here? Some technical terms we are to discuss. Any deviation or disturbances from the healthy condition which interferes with the normal and performance of the vital function of a plant is called a disease. So what is the here symptoms? The external signs. So that is the morphology, external morphology on the plant which is indicate the disease called as the symptoms. So disease may affected that state the whole plant or either systematic disease or sometimes some diseases affected contain organ or the host or parts of those organs is called as a localized diseases. So entire plant is affected by these pathogen that is the systematic diseases are there. So any particular plant, any particular part or organ which has affected by the pathogen that one we call as a localized disease. So these are the certain terms which is will be useful and that is a blah explained in these causal agents. What are the causal agents? These organisms so, which cause the diseases in the terms of casual organisms or and the parasite. So, an organism which is lives upon their living organic matter, such as tissues of plants or animal, is called as the parasite. A parasite may or not all always cause the disease. So, what is pathogen? A living only which cause the disease is called as a pathogen. Inoculum. What is the inoculum here? Some technical terms we have to discuss. What are the inoculum here? Wait. So here inoculum is what here inoculum and so the inoculum is the disease causing organism only survive from one growing season to another in some forms such as the species are sclerotia and dormant mycelium in the various other forms in the and the host seeds wild herbs growing in the vicinity and remains of the organic matter of the diseased plants of the previous season. Any form of infectious and agent, whether a dominant spore or sclerotium or dormant mycelium, that constitute the primary mycelium. And the source became the so that is the source of primary inoculum. The organism may produce a fresh crop of infective bodies on the host plant, which serve as a secondary inoculum or and bring about the secondary infection. So penetration, the prior to infection, the casual organism land on the host resumes activity uh, that is the germinates again entry into the host directly or indirectly. This invasion of the host tissues by the living microorganism called as a penetration. So infection and incubation period. Yeah, so incubation period and disease cycle. Disease cycle means a series of several means some between the infection and complete development of any diseased construct is called as a disease cycle. So here one by one we are to discuss classification of the plant diseases. We are to here abiotic and biotic parasitic fungal they are to discuss. Okay. So the plant disease may be classified into two main categories are there that the nature of the agents that is the abiotic or the biotic agents only. First one is the abiotic means non-living or non-parasitic or non-infectious diseases. These are the introduced by the unfavorable conditions of abiotic factors such as the soil or air, which is include the temperature, moisture, nutrition, and toxicity. So, and some cultural malpractices, that is the common example of the non-parasitic diseases include the blooms and rot of the potato and black heart of potatoes. These are the some example of the abiotic. So biotic, another one is the parasitic or biotic infectious diseases. These are the caused by the attack of living agents that called as casual agents. So, so these diseased plants are called as host. That is the example of the biotic cause agents. Plants include the fungi, fungus like organisms and slime molds, bacteria, mycelium, and the spiroplasma, phytoplasma. These are the some diseased agents are there. So now coming to the paddy. So paddy blast diseases here I shown to one diagram. This one is the paddy blast disease. So platyblast, that is the organism is the Mangaptera oryzae, that is the Pericularia oryzae. Another name is we call it the Pericularia oryzae or Mangaptera oryzae. So platyblast caused by the fungus Mangaptera oryzae is one of the most devastating agricultural pathogen in the worldwide. So it is causing to lost their yield. So here the plant disease is one of the most served disease affecting the paddy and is prevent the all over the world. It also known as the rice blast, paddy blast or rice blast. So the rice rotten neck and rice seedlings, blades and blast of the rice or rice fever. It is a major problem in the rice producing the commercial like Japan, India, and USA. The blast is the most served in area with the high humidity and rainfall. Affected all the above the pranahita means effect on the ground pot or the rice plant, leaf and leaf blade and leaf sheath, stems, collar, node and neck parts of the panicle, seeds and even roots also. So in India, the blast disease is more common in the southern part of India, particularly in the coastal area, and loss due to the disease maybe up to 90% of the crops. So that is a member of the Magnoptera gracile complex structure. So, so it is caused the blast disease, blight disease, inserve the cause the infectus entire the crop gives a blasted appearance. Hence, the call is the blast disease. So symptoms. The disease can be infected paddy and grows stages are aerial part of the rice plant, that is leaf, neck collar, and neck nodal area. Initially, the symptoms are seen gray or brown lesions and stop with the narrow broken and produce on the all parts of the plant body. So this diagram is the blast diseases. So small pieces. Okay. So these are the some symptoms are caused by the blast diseases. Okay. So another diagram or picture I shown. So this one is also there. So here I shown that diagram. Wait, 1 minute you wait. Just I express another figure. So this one is the so some symptoms. What are the here? The small packets originated on the leaves, subsequently elongated into spindle-shaped. That spots may be spindle-shaped in structure. So that is the so spindle-shaped structure is appear and what here wait. So with the ashes center. The older relations are elliptical or spindle shaped and whitish to gray with the necrotic borders and several spots collations to form the big irregular patches and kill the entire leaf. And the nodal infection cause the column to break at the infected area. The inner node The inner nodal infection also occur at the base of the plant, which cause the panicle similar to the introduced by the yellow stem borer or water deficit. So lesions on the neck are grayish brown and cause the giddling of the neck and panicle to fall over. So that is the some infections and another Casual another organisms are there. That is the pathogen disease rise in pericular oryzae. That is that is the perfect stage of the pathogen in the Magnaphthera. So mycelium is the septate unicellular and or uninucleate branched and icilium and hyaline to alveolus in the color. So the condition conidiophore develop a single or in cluster through the stomata. There are the namely solitary or branched two or four septate with the swollen base and tapers toward the lighter apex. So conidia are piriform condition to uh, obtuse in the shape with the rounded base and tapering. Okay, so that is the conidias are there. I showed the diagram. So these are the conidias. So here conidia 4 is there. So this is the blast disease pericularia oryzae. There is Magnaphthera, the perianth structure, and these are some conidias are present. So this is the cycle, life cycle of the these blast diseases. So rice blast fungus, there is the 14 stages is developing on the can and stem, nodal, and panicle. And foliar infections is mainly caused by the that is the attachment of conditions to the rice leaf cuticle. So a single cycle can be completed about a week under the favorable conditions, where one lesion can generate up to thousands of the spores. A single night of disease lesion, however, can spread 2 or 3 days after infected. With these with the ability to contain or continue to produce the spores for a around 30 or 25 to 30 days, rice blast lesions can be devastating or susceptible rice crops. So this is our life cycle. I shown to the life cycle of the these so discussions. So here, okay, clear. So I shown to that diagrams. So Okay, so that one is the right. Wait, 1 minute. Wait, we have to share some videos or means diagrams. So, so in the this is the life cycle blast fungus attack the rice plants at the all stages of development can infected leaves, stem, nodes, and all the here panicles also. So wait, I shown to the diagram. So is not give the most. So oh, the Rakesar is not give the Shravan or Rakesar is not give the coes. So that's why I cannot to shows. So I cannot to share. I'm not accepted to share in this. Means pictures or whatever it may be. So whatever it may be, we will continue. So in a single cycle can be completed in about a week under the favorable condition where the one lesion can generate up to thousands of spores in his one night or single night. So here the disease lesions, however, can appear in 3 to 4 days after infection with the ability to continue to produce the spores for over 20 days. So rice blast lesions can be devastating to susceptible rice crops. So this is the some control methods. How we have to control the mm, so wait is a picture. Okay. So here I show to um, so this is the life cycle of uh, the blast diseases, right? So how we are to control these blast disease? Here mechanism the separate the viable spores into the crop residues from the newly emerging seedlings. That is the remove that is the collateral weeds pushed from the bonds and channels. And sanitation is most important. The cleaning of the plant debris from the field residues and the sources of primary inoculum. The debris we have to remove and use high quality and disease resistant seeds we have to use. And what is the chemical treatment? Means chemical treatment that is the most important. That is the seed treatment. The pathogen is the born on the head. The treatment done in the agrosum or copper sulfate or fungus. These some fungicides like carbendazim is also there. That is the tetracyclose and uh, azoxystrobin. These some pathogens means fungicide treatments also we can do. And foliar sprays means copper fungicide mixed with the mercury compound and phenyl mercuric acetate is highly effective on the foliar spray. So next one is the brown leaf spot. So brown leaf spot, that is the oryzae, that is known as the helminthosporosis. So helminthosporosis disease is distributed in the almost all the rice-grown tracts are world, but it is the occur most severely in the southern East Asia, Japan, and Philippines countries, and southern part of the USA. So in India, disease was first reported in 1922. It is reported to cause the damage to rice crop in the eastern and southern rice growing regions of our country. So besides causing the heavy leaf spotting, that is the severity of the disease may reduce yield when airs may be sterile and gain the formation of the spotted. The diseases cause the great economic loss. The infected seeds have very poor germination rate. So that is the symptoms. So okay, that is the brown spot diseases. I shown to brown spot diseases. Please wait the students here. I shown to that diagram, that picture here. We can explain these brown spot diseases. This is some brown spot disease on the horizon. So what are the symptoms here? Pathogen usually attack all pods, rice plant at the stage of developing on existing roots also. These may be complete failure the seed germination, or the seedlings may um, succumb to the disease before they emerge out of the soil. Most conspicuous symptoms of the diseases is appearance discrete or dark brown and ellipsoidal or eye-shaped spots on the upper surface of the leaves. The spots often vary sizes, shapes, and appearance are there in the form of very small brown area surrounded by a yellowish halo or gradually increase in size, turning to dark brown. So along with the increases in the size, the spots also increases in the number of very rapidly. A fully developed spot has the grayish brown central portion surrounded by the deep, so reddish brown or margin, and maybe big 4 to 6 diameter appear. Okay, so it I shown to that. Spores, conidiospores also. I show to conidiospores. Here, spores. Mm. So these are the spores are there. See here, brown spot of diseases. So the cause here, the case of serve the attack and ears failure to emerge from the sheath and perish without the further development. Lesions on the glands causing a brown or discoloration of the canals inside. The quality of the grain is also affected in case of serve the infectious on the grain formation takes place and here became a sterile. So here, so casual organisms. Here the casual organisms pathogen is the Dracharella oryzae or Subramania. This is called here Helminthosporium oryzae. Helminthosporium oryzae. The fungus produce septate, prostate, and much branched hyphal. So hyphae grown both the inner and intracellular in the host tissues. The conidiophores arises as the lateral branches from the prostate hyphae. There are the erect, short, short, septate, or unbranched exception at the base, where they form branches toward the rapid region. They are the bear the. So cylindrical left or straight or slightly curved and slightly uh, tapering toward the rounded end. So smooth walled brown colored and vary greatly in the size. Here I am shown to some spores. Here wait, I shown to this diagram. So this one. So this is the some conduit force breaking the conidia here, and these are the. So this is the some conduit forward development. So this is the cycle. Brown spot rice is located. This is localized diseases that the pathogen is restricted to the infected zone and cannot pass long distance through the inner tissues. The buildup means inoculum from the infected region of disseminates further and cause infection. So brown spot of rice is located diseases and pathogen is restricted to the infected and cannot pass through the distance through the inner tissues. And the buildup inoculum from the infected region and disseminate further and causes infection. During the germination of infected seeds, the coleoptile gets infected and then gradually it affects the seedling showing blight symptoms. The infected regions of seedlings, that is the coleoptile leaves to produce conidia. The conidia are often dissemination by the wind or rain fall on the different regions of the same seedling of different seedlings and causes the infection. So this one is there. So how to manage these brown spots diseases? So there is a cultural method. The eradication of collateral means the germinations or rice field weed host sanitations and irrigation of water, leaching of metals. Irrigation of water means during the irrigation time is also should take care. Should water not pass from the infected field to the field without disease. This one is the most important. Irrigation system is also and leaching down the metallic elements like iron, magnesium, and calcium from soil damage. They should be leached. Otherwise, the plants became more or susceptible to the disease. So applications of metals here potassium and the field during the growth period and spraying. So another one is the nitrogen fixation. So nitrogen fertilizers means optimum nitrogen fertilizer application transmitting and optimum dates provide to the helpful for the reduction of the diseases. So here physical control also sunlight, hot water treatment, cold water treatment. That one is also. So chemical treatment means seed treatment, dry dressing treatment, and wet treatment, foliage treatment. We can use it. Mm. Here foliage of seedling rice. This one is the most means that one we call as the BOTANY disease. So Physicorum physiocoria. So Physaриum physicoria. So there is an important disease of rice occurring throughout the rice or is the sativa. Physaриum physicoria. The Polish seedling diseases are fast root or Physaриum blight. That one we call as. So the disease was reported for the first time in India. Um, so a metabolism of the fungus produced a surplus of gibberellin acid, which is a smooth hormone, and causing the hypertrophy. Also, effect is they shows abnormal allegations and serve these gibberella fusarium correct. So here, so block or stem rust on wheat. I show to the diagram also. Wait, you just few minutes. You have to wait. This is the so rust on the wheat. So black or stem rust on wheat. Okay. So diagram is clear here. I shown to that diagram is clear. So this is the blast or rust disease on the wheat. So disease symptoms are there. What is the here? The wheat growing countries of the world or in India appear the different times of the years in different parts of the country. It appear in the month of the March and November in India. In the southern and peninsular India, it appears every year in the November. So the stem rust is the serious threat to the wheat in India. It causes serious damage in the moist areas and moist seasons. So what are the disease symptoms here? So elongated pulses or stecks are produced on the leaf or stem, on the leaf beard or even on the glues, which is uridia, sorry, or uridia. So, so each undersorus contain the most of the redly brown. Uh, so here, these are the uridia and iridosaurus. Each iridias contain the mass of the reddish brown, rustly red one called that is which is the binucleate. So under that's the iridospores we have to call. So these iridospores are produced by the dikaryotic or secondary mycelium in the parasite sting and tissue of the virus. The spore stage of the Parasitic is called as a red rust or summer stages are. So this is the some diagrams. I shown to the some iridospores. So these are the iridospores are there. The okay. So these diagrams here iridospores. Symptoms on the barbary host. So here two host tissues are. So infected barbary leaves shown that is small circular, slightly elevated yellowish or orange spots on the upper surface. So each spot is denoted with the uh, indistinct pump as astiole of the sporangia. So a sticky liquid being the exuded through the astiole on the leaves side of the leaf cap shaped is acid or acidial cup they are produced. They are usually appear and opposite and circumferential uh, mm, pycnidial spores. The axial cup are oranges yellow in color. Each one of them conditions mass of the spores and they were binucleate we call as acidiospores. So here on the acidiospores, I shown to the diagram. See here. So this is the so this one is the lower area, lower surface area. This one is the spores is appears. So what is the casual organism? The casual agent that is rust disease, the Fucsinia graminis or tritiantum. So here mm, hetero heterocleus parasite. So which is completing the disease cycle in two hosts, that is wheat and another one is the barberry. Two plants they are to need to complete their life cycle, that is the wheat and another one is the barberry. So that way heterohostaceous parasite we can call heterogeneous parasite. So disease cycle here want the tetraspores are covering the organs, but in the plants in the India under spores condition, there are carried from the distinct high altitude and hill and wing often the so each cell produced in the a shorter promycelium into divided into the elongates and so each syncytia undergoes the meiosis in the um, promycelium are the so walls and laid down the haploid nucleus. So the promycelium or epiblast became the septate and form the single cell. So each cell of the blastoidium. So these are the some diagrams. So wait here, I show to that diagrams also. So this is the Puccina graminis. So Puccina graminis that is there. So here loss of smut on wheat. So here the loss smut on wheat is a district 2 disease. The disease is very common and widespread due to the characteristic black dusty appearance of the diseases. Inflorescence is also called as a black must diseases or the back head. So disease is more abundant in the humid and semi-humid regions. So cause the here there is a great damage in the wheat growing area of India, particularly in Punjab, Uttar Pradesh, and certain districts of Madhya Pradesh. And so, what are the symptoms here? So, symptoms so that is widely visible only when the plants develop area. The infected plants generally all are the here all these spicklers and kernels of each year the snurs the plant cells. So here the spicklers are affected. The ear get damaged in the dry powdery mass of spores they are to generated. So here loose spots diseases I show to the diagram. So it this is the way. Okay, now okay clear. This is smart disease. So here disease cycle the mass of the chlamydospores from the infected air are disseminated by the wind or any other agencies to the healthy or cylindrical adjacent plants to the flowering September and November. So dikaryotic dikaryotic phase is established by the fusion of germ tubes that is a derivative from the individual uninuclear cells that from mycelium and ultimately infected hyphae developed. The infectus hyphae penetrate the young tissues at the base of the ovary. Along with the when that is the above process, the fungal growth and the pollution and infectious and imply function. The profuse mycelium is present in the scutellum. It mature infect of the hyphae. Most of the infected grains cannot be morphological distinct, effect showing the infected seeds developed seedlings and non-tendons. Here, there is a great here. This one is the psychology, means life cycle of the shown to the life cycle of the this diseases. We wait some time. So another disease control method is there. What here disease control method only? What here the physical method, hot water treatment. The seeds will be treated with the hot water treatment only. The seeds are soaked in the water around 30 to 40 degrees for 3 or 4 minutes. We activated damage the and mycelium to germinate. So and soaked and seeds are quickly transferred into the hot water in 10 minutes to kill the internal mycelium according to the journal. So these immediately fungus could be completely eliminated by the soaking in seeds in the hot water. So solar energy treatment also nowadays we can use solar energy treatment also. So water soaking treatment and chemical treatment here dry dressing treatment, that is the carbon dioxide and binomial and carboxyl we can use. Baviston powder is also we can use. And biological methods here TB5 and trichoderma strains, trichoderma viridi we can use it to remove these diseases. So hot water treatment also nowadays we have to apply it. Hot water treatment here significant cannot control graduate by the soaking the seeds in water containing this is sodium hypochloride or so this is tolerant so here we can IARI means Indian Agriculture Research Institute. They have to produce the here in New Delhi and Kalyan PV18C32 WG 307. These varieties, this is resistance varieties we can use it to better to control these diseases. Okay, thank you, Andy. Thank you so much for nice presentation. Tomorrow we'll continue the next session.